Okay, for the sake of punctuality, let's go and start. Hello, everybody, and welcome to this workshop about BiaPy as part of the Halfway to I2K conference. Today, the presenters will be Daniel Franco-Barranco, and I, who are the main people responsible from BiaPy, which is this software that I will introduce to you in the next half an hour or so. That is our tool to create what we call accessible deep learning-based workflows. Okay, so just very briefly, the outline of this hour would be like this. We'll make an introduction to BiaPy, led by me. I will describe a little bit what a workflow is, so we understand by a workflow in a common bioimage analysis setting. A brief recap about deep learning, because BiaPy is all about deep learning, and what we can do and we cannot do with BiaPy. Okay, and then we will describe a few workflows that are available there in our tool. And then for the second part of the of this workshop, we'll go for demo time with Danny explaining us how to exactly do one of these workflows for a specific task, which will be to the instance segmentation. Okay, so first things first, what is a workflow? And for that, I always quote my friend and colleague, Ko Tamura, who says that the workflow is nothing but a sequence of image processing algorithm methods, no, with a specific parameter set that we use for a task, no. In our case, we have a problem. This is a general description, no, where we have some inputs. In our case, they always images of any type, as we will see, and usually they don't come in a proper way, so we have to pre-process them, normalize them, to do many different things, so we can pass them to what we call the solving algorithm. The solving algorithm, in our case, in BiaPy, as you will see, is always a deep learning model. You can do image processing without deep learning, but if so, BiaPy is not your tool. And then the result is usually a little bit raw. You cannot directly analyze it. So usually you have to post-processing. There is some algorithms you have to do. You have to maybe merge some of the results or of the model, etc., to do the proper analysis and get your actual results. So quick recap in deep learning. Deep learning is this subfield of artificial intelligence, which is, as you know, a very, very hot topic nowadays, in which what we do is that we teach the computer how to do a task without explicit instruction. And how we do this? We do this by showing more and more samples to the model, so it learns to do the task. The typical example, especially in image processing, when we this is the one that we do very often in computer vision, that I teach to my students from the very beginning, is this example of a data set of images where the main content of the image is a cat or a dog. No? So basically what we do is that we show the an input image to our model, a deep learning model, and with some operations that we're not gonna define now, the model spits a prediction about the main content of the image, if it is a cat or if it is a dog. And then there's a step of evaluation. We have to decide if the actual category of that image was correctly predicted or not. In this case, it was. And then what we do is that we iterate and we show more and more images to the model, and based on how good it classifies them as one class or the other, we correct the parameters of the model so it gets better and better at making those predictions. So when we work with images, the most typical case is that for what we call the training, okay? And then once the model is trained, what we have is the test phase, where we show the model without any changes. So we call this a frozen model, new images of cats and dogs, hopefully, And then the model has to do exactly the same task. Just predict if those unseen images that haven't been used for training are actual cats or dogs. Okay. When we work on deep learning, especially with images, what we have is as input always an image. When we work on bioimage analysis, it could be of different channels, different dimensionalities, etc. And then usually the first part of the model is what we call the encoder. No? So the model goes extracting features at different scales, and it goes summarizing the information and reducing that information in terms of resolution until a point that we usually call the bottleneck, where it has to go up. This is sometimes not necessary. And then it goes recovering the actual size of the image. Okay, depending on the task, we have only the encoder, or we can have also this decoder, as we will see. And then it produces an output. Okay, in this case, if the input is so we have an input image, and the output is of the same size. So we use an encoder and the decoder. And whichever the task we do, at the end of the day, we have to do the same thing as I explained before. We have to check if the prediction, either by pixel or by full image, was correct or not. And then, based on this comparison, do the correction of the parameters of the model. Okay? Good. And this is actually something very, very useful in bioimage analysis. As you probably know by now, we can do many different things to detect cells, their positions, their locations, or different types of organelles in many different types of image modalities, to follow them in time, to detect specific objects of interest in I don't know, at the scale of a full organ, etc. So at this point, you may have this question. Okay, I heard about so many different deep learning libraries. So why do you want to just create another bioimage analysis library based on deep learning? Isn't this something that is already solved? And actually, this is something that I also ask myself, especially when I'm in front of, for example, in a conference or a new paper that is showing a new tool that is perfect for my data, but then I have to ask the right questions, which are, okay, is this new tool something that is actually accessible, portable, flexible, and reproducible? And many times the answer is not that fast. Okay, so why are these questions important, and what do they mean? Actually, when I mean accessible, I mean that if you create these pipelines, or workflows to solve your image processing problem, then the users should be able to deploy them from any computer environment, and this is very important, with minimal time and effort. They don't have to basically start from scratch if they want to do it in a different computing environment. They should be portable, meaning that the user should shift those analysis pipelines between computer environments once they are developed, and that should be simple. They also should be flexible, of course. We would like to have those pipelines customizable, meaning that they should not only work for the data of the paper where they were published, they should be able to work on other data sets with some tuning, of course. And finally, call me crazy, but we work in science, so they should be reproducible. So running the same software on the same input data, we should obtain the same result. Okay? And unfortunately, this is not always the case. This is not something that I made up. Actually, these 4 key questions here are actually from a very similar community to ours, from the neuroimaging community that they published last year, this very nice paper called Neurodesk, addressing these problems in their community as well. Okay, so here's how we started BiaPy. It was finally published this year in Nature Methods as BiaPy accessible deep learning on bioimages. And let me show you an overview of BiaPy. In this platform, what we try to do is to address the full community of not only users from the very inexperienced users to the most experienced users or developers, okay? But also would like to have a library that is specialized, even if it is using deep learning, on the type of images and data sets that we have commonly produced in bioimage analysis, that are usually multi-channel, that they have multiple dimensions, and they can have, I would say, a large diversity in size. They could be just a few megabytes, or they could be terabytes of data set that we have to handle. And for that, we also target a few analysis tasks, not all of them, okay? So we will see, we have segmentation, we have detection, etc. Sorry, no, somebody said something? No. Okay. I hear anything. Okay.
Yeah. So there was someone, Philip. Can you repeat the question, please? Philip, are you there? You were about to
maybe
ask something
maybe
to Ignacio? No, okay.
Maybe he was talking somewhere else. Okay. Anyway, so we have a few tasks that I will describe later, but we don't do everything. For example, we don't have visualization, we don't have registration. For that, we have very nice tools already in similar ecosystems out there. Okay. Regarding the backend, okay, everything is as nowadays in Python, and also our let's say core deep learning library is PyTorch. So this allows us to, on the one hand, use all the models that are already pre-trained in the Torch Vision community, but we also can, as you will see, we're integrated in the bioimage model too, so we can also share and consume models from the zoo. And of course, you can produce your own models and train them directly in BiaPy. And for the front end, as I said, we are actually targeting many different type of potential users and developers. So we have a very long range of possibilities here. From the most non-expert users, we have a graphical user interface with a wizard that is actually making you natural questions to try to understand your problem and then define in the background the kind of workflow that you need, and it produces all basically the instructions that you need for this. Okay, we can also now, and this is a novelty, run these workflows in Galaxy. You will see when we basically achieved this a couple of weeks ago. We have notebooks, Colab notebooks with no code that you can run either in Google Colab or locally. Everything is installed, and this is quite interesting, in a single Docker container. Okay, so you have, as you will see, all the workflows that are available in BiaPy with a single installation. Okay, and then of course, if you are a hardcore developer, you can run everything in the terminal or even integrate the code of BiaPy using our API. Okay, so how does it look? As I said, the core of BiaPy is the workflow, and the structure is the same as I showed you before from Kota. No? We have basically divided our platform in 3 different modules. We have pre-processing, the deep learning model to do either training or testing, and then the post-processing model. Okay? As input, we always have images, but they could be also come in with some labels depending on the tasks. They could be the mask of your objects, or they could be the categories of every object there, or the content of the image, etc. Or even a CSV file, I don't know, with the locations of the objects that you want to detect, or the categories, etc. And the output, it depends on the task, but it could be an image. For example, you have the input image and the mask image, or the super resolved image, or the denoise image, etc. Or you could also have a CSV file with some of the could be locations, categories, etc. So in the pre-processing model, we incorporated everything that has to do with preparing the images for the deep learning model. Okay, so usually you have to do patching, no, dividing the image into pieces, do some normalization, etc. In the post-processing model, we incorporated everything that has to do with making sense of the output of the deep learning model, because as we said, it's usually a little bit raw, but you have to also prepare it a bit for the final analysis, or you have to redo the full image out of the pieces, etc. And we did it this way because on the one hand, we think that for users, it's very easy to just change some of the parameters of the workflow and then combine different pre-processing, post-processing methods or models, okay? And also for the developers, it's quite straightforward to just go and contribute to any of these models, no? They don't have to contribute with a full workflow. They can just decide on use a new model or a new reprocessing method, etc. So under the hood, whichever your front end is, everything gets stored, the full workflow, in a single text file, which is a YAML file, which is where we stored, on the one hand, where the data is, so very important, no? And what type of data. And here, it is relevant to say that we support different typical formats, but very importantly, we support ZAR and H5 formats, which means that we can load into BiaPy datasets that are larger than the RAM memory of your computer. So we can actually scale these methods to very large datasets. Then we have everything that has to do with the different steps of the workflow, the pre-processing, of course, the training or the testing of the model, etc., and the post-processing as well. And I usually would like to say that this is done in a quite memory efficient way, and also taking advantage of all the resources of your machine. So if you have multiple GPUs, you can run it in multi-GPUs. You can run it in a single GPU, and of course, if you don't have a GPU, you can also run it in CPU, which the training could be very, very slow, but for testing, it could be enough. Okay, and of course, at the end, you also define in this YAML file the output that you're gonna get, no? The different type of, for example, metrics that we're gonna apply for the analysis in 2D or 3D, and what everything gets stored. So at the end of the day, if you run a full BiaPy workflow, and then you want to share it with someone, the only thing that you need to share is this YAML file, which is a very small text file. So this other person can reproduce that in their computer. Okay? And if you want that person to reproduce exactly what you did, you can also share the train model that you use in your workflow. So it's another file, not too big, probably, that you can pass to your collaborator. So how do you configure a workflow? Actually, a workflow looks like this. So it's just, as I said, some text, some keywords. Here are shown in blue with the entry to those keywords. So in this case, this is an actual workflow that we use in one of our papers. We have the first part where you define where the data is, the type of problem that you have, in this case, semantic segmentation in 2D, the size of the different patches that you're using, and very important, where the data is, okay? And then we have everything related to the deep learning model and how you're gonna train it, in this case, what kind of type of architecture, the optimizer, blah, blah, blah. And at the very end, just how you're gonna post-process that data, okay? So the main idea is that for every type of problem, we have a template with these keywords, so the user can only has only to change those entries. The advantages are that it's very easy to prototype. You don't need to code. You just need to change the text file. But of course, you can say, yeah, but wait a minute. I don't know all those keywords. I don't want to go and run into the documentation to see what every keyword means. And honestly, I don't know all the keywords either, no? Because in the end, what we usually do is that we produce those text files through a notebook or with the help of the wizard. So you can do it in a more human way. Okay, so if you want to know about BiaPy, there are two main pages that you have to visit. Okay, let me do it fast. If you go to the BiaPy github.io, see if I can do it. Yep. Then this is the what we call the landing page. Okay, so you have access to the documentation, the GitHub repository, etc. And here you select the front end that you're gonna use and then how you're gonna install it. You have the WE, the notebooks for every different workflow, everything about the Docker container or the command line with different options. Okay, and then the documentation, either you go from here from my presentation, or you can also click here. Okay, and then you have everything from the different workflows in this typical format of read the docs.
One thing, please keep these 2 pages open. So because we are going to reuse them in the demo, okay? Just to let you know.
Okay. So as I said, you can just type in Google BiaPy, and then it will probably bring you to this landing page. Okay, and then you have the options here. And if you go to the documentation, then it would open this other image, this other website here. Okay, so let me continue my presentation. Okay, so what are the typical tasks that we do in computer vision, no? When we have this type of deep learning models? Well, a classic one is what we call semantic segmentation, where the input is an image and the output is an image where every pixel has been classified as belonging to a different class. No, in this case, the pixels of the dog are classified in red, or with a specific, this is just a color map, no, with a specific number, or the pixels belonging to the jeeps, they are in blue, and then probably the background will be put to 0, black, or whatever. Here we put it in transparent so you can actually see the mask. This is something that we can do in BiaPy in 2D and 3D, for example, to do a segmentation of organelles, right? In this case, this is just binary segmentation at the pixel level, okay? These presentations will be made public, so you have all the links here to do everything as well. Another, let's say, type of segmentation that is a little bit more complex is what we call instant segmentation. So what we do is that it's similar to the one that I showed you before. Okay, so every pixel gets a different ID, but now all the pixels of a single object belonging to the same category get a different ID. Okay, so all the instances of the different classes, now in this case, there were 3 ships. Before we couldn't differentiate them, but now we assign them a different color. Okay, so this is what we call instant segmentation, and we have ways of doing this in 2D and 3D, multi-channel, and in BiaPy, for example, with big data sets. Example to do, and this is similar to before, no? Mitochondria segmentation on EM images, but now for all the instances of the mitochondria. Then we have in computer vision a typical problem we call object detection, where we try to find the different object of interest on an image, and usually in natural images, what we do is that we find boxes, we call bounding boxes of the different objects. They could be overlapping, no? And then we can we differentiate the different ships with a different box. Okay, but in usually you want to export this to 3D, no? Doing boxes in 3D is more complicated. So what we usually do, in bioimage analysis is to, instead of using boxes, go and get the center of mass of every object. For example, as in this example, to get the center of mass of each neuron in a full data set. And this is easily expandable to 3D. Then we have the problem of image denoising, where we have noisy images that we want to basically clean, and the output is the noise image. So we have nice algorithms to do this in an unsupervised way, so you don't need to have pair noisy and unnoisy images to train the model. We have what we call the problem of single image super resolution, where you have in natural images, it looks like this. No, you have images that you want to improve the
Nacho,
quality.
one
Yes.
thing that there is a question.
Mm-hmm.
So you don't have detection with bounding box in 2D? No, we don't have.
No, we don't
We are
support
not
this stuff.
using bounding boxes. No, we tackle the problem of detection, but by predicting the center of mass.
This is something that it could be directly extracted, for example, from instance segmentation. You can get the boxes. It's something that is related. But and in instance segmentation, we actually sometimes also detect the center of masses. But we don't use boxes for that. Okay, because we want to use the same workflow or the same idea in multiple dimensions. Finally, we have this. We have super resolution, where the input image is a low resolution image. Okay, usually it could be low quality, and then the output image would be a super resolved image with a better quality and sometimes bigger size as well. No? It could be the same size with better quality, or it could be just, or it could be also bigger size and better quality. No? And this is something that is typical problem also in bioimage analysis. In 2D, it's also typical to increase the size of the image. In 3D, usually just do same size but better quality. All these options are there. Then we have a specific workflow, which is a little bit special because the output is not something that you can use straight away in your images, which is a self-supervised learning workflow. What you do is that in general, you have images, you pre-train a model by doing some task. For example, here we mask parts of the image, and the model has to reconstruct those parts that are missing. Okay, so you can do this with data that hasn't been labeled. It has no mask, no detections, no categories, etc. But it allows the model to learn about the content of the images. Okay, and the output of this at the end is a train model that you can then use as a starting point for any other task. And for example, you can do denoising of images, then you get that model, and then you can just retrain that model for segmentation with just very few labels, and usually works better than starting from zero. We have also image classification that, to be honest, we don't use that much in BiaPy because this is something easy to use with other libraries, but just for the sake of completeness, we have 2D and 3D classification of any kind of images. Okay, and something that I think is more important is our final workflow, the final acquisition. That is this workflow to do what we call image-to-image translation. In natural images, this is something that you use, for example, to add color to an image, or to do from a sketch of something, create an actual image with generative models, or for example, from labels, you create the original image, etc. But in bioimage analysis, this is typical to do what we call virtual staining. No, for example, you have a bright field image, and then you can produce automatically the equivalent stain channel for the nucleus or for the membranes or whatever you have the training per data prepared for. Okay, so that's all for me. You have questions so far? You can interrupt us now, or you can do it interactively while we continue with the workshop.
I have
Okay.
a question.
Yep.
So, yep. Sorry. Go
Sorry, I
ahead.
have a question for the detection of the centroid problem. Could you also kind of combine this with a specification where you kind of maybe classify types of cells or something? Is that done already?
Yeah. So not only detecting the centers, but also classifying the cells that you're detecting. Yeah.
Yeah, so this is just an option in our workflows. So you can have the coordinates of those objects and their categories. And then we have workflows and models to do that. Yep. Very good question. Something else?
And yeah, also, sorry, a second question.
Yeah.
For this image to image, so it's like the example you showed is more kind of like, okay, you have the larger cell and then you go to nuclei. So I would be interested if you, for example, have a nuclear stain and then a bright field image which has, like, it looks like visually looking at, you cannot say where a cell starts and ends. Could you kind of use these both, or was there something like this done already where you can then predict from the kind of together with nuclear state and bright field to like a kind of cell reasonable like cells segmentation?
Yeah. So this is something that that actually this workflow was prepared for a challenge. Well, we have the input images were like this, bright field images, actually with different zooms and they were not always how do you say this?
Yeah, they were like out of focus.
Focus, or
Yeah.
yes, that's it. They were not focused always in the same places, and then we have multiple channels with different organelles, no? And that was the task for the challenge. But that being said, something that people do a lot is if you have, for example, the bright field image, then you can do something like this, then produce the nucleus and maybe the membranes, and then do segmentation on those with another model that knows how to segment that data better. And then it's a good approach to get those segmentations from bright fields that, as you said, could be quite noisy and the borders are not actual borders are very clear.
Okay, thank you.
Yeah, I mean, because these workflows you can apply like one workflow after each other. So once that you do this image to image task, you can like apply the segmentation task afterwards.
Yeah, something that I do, for example, with my students, I'm teaching on a computer vision class for master students, and then we do exactly what, as I said, we train one of these models to create the nuclei, and then we use that to do the segmentation on the actual images. So it's much simpler because the membranes are usually better seen, and then you can use this as the seeds for the segmentation. Okay. If there's no more questions,
if you have more questions, please go ahead. No? Okay.
Hello, I have a question.
Yep.
So, 1 second. I saw in your documentation that there is an option in the instance segmentation to select different models.
Yep.
And it seems that there are mostly various types of UNet. Is there any plans to, in the future, add something like SAM, SAM2, or microsam or something like that directly into BiaPy?
No. I mean, we have more than units. We have transformers and other kind of models too, but we are not planning to add any kind of model based on SAM because you need prompts. I mean, this is like an automatic pipeline, right? So those models are
has an option of automatic instance segmentation as well.
Yeah, but you need to know the object shape. I mean, and if you want to do like 3D, you just reuse the same point in all the set slices. But whenever you are like out of that object, you are going to segment
actually,
something that is not the object. So,
if I may interrupt, yeah, in Microsam they also have now the option, but based on a unit, to do the automatic segmentation. Very briefly, we don't plan to incorporate Microsam. We see each other as complementary, But what we do have in preparation is to use strategies such as cell pose, stardist, that we can go straight and apply that work very well for this type of images, and then reuse the full structure of BiaPy to, for example, run it at a big scale. No, this is something that we're exploring to, because it's simpler, as Danny said. If you want to apply this in a generic way without knowing anything about the images, to create different strategies based on how you represent the instance segmentation problem. Okay.
Yeah.
And the
So
other question was, when you talked about the unsupervised learning, what backbone do you use there? Because if I think I recognize the image that you used with the flamingos is from the Mask outing club.
yeah, yeah, you're right. You're right. Yeah. We have two options there to pre-train the model. One is like modifying the input image, like to add in noise and crapifying them, let's say, to record. But also we are using the mask autoencoder approach. Yeah.
Okay, cool. Thank you.
Yeah. And what we are planning also in self-supervised is to add a work that is Spark. It's to pre-train not only transformer but convolutional neural networks in the same way of masking the things. And also going back to the previous question, we are not in our plans there are no plans to add this microsum or foundational models in that sense. But yeah, we want to add like use Dino V2, Dino V3 weights, so we can like in the same what's the last work of Cellpose, no? They are reusing the weights from Dino to do the segmentation
Yeah.
task. So we are going to do that in the future also. Yeah.
Okay, cool.
Okay. Any other question? Okay, let's move on then. So this slides is just to present you briefly to show off maybe a bit of the things that BiaPy can do. So this is an instance segmentation workflow. So you can, as Nacho has explained, you can predict instances within small cubes. Okay, you can scale it a little bit by, I don't know, regions of volumes, okay? And you can also do with BiaPy something like this, which is like extracting all the nuclei in this case in a Zebrafish, okay? So I mean, depending on your specific task, BiaPy is suited for handling small, medium, and large data scale, okay? So I want you in this part to emphasize that, as Nato has explained, okay, so BiaPy can be run in different ways, as Nacho has explained, but here in this demo, we are going to just depict or show one option, which is running through Google Colab, okay? So if you have the presentation there, you can open this link with me, or maybe you can access also the notebook that we are going to use in this demo by going to the landing page. This one. Just tell me if you want me like to go slower. Okay. So go to the landing page, and then in collect notebooks, here we are going to play with the instance segmentation 2D notebook. Okay, so if you open it, so in this demo we are going to try to go like step by step on how to resolve an instance segmentation workflow. Okay, so as Nacho has explained, this instance is about identifying uniquely the objects within an image, okay, with different IDs. In this case, the mitochondria are depicted here, are segmented. Okay, so if we go, this is like the overview of the task that we are going to solve. Okay, so if we go down, there is here an explanation of the inputs, the expected inputs and outputs that this workflow is going to needs and it's going to produce, okay, and how you might need to structure your data in case you are providing your own data, okay. So this notebook will be like cell by cell. We can like run them one by one, okay. So if you do it with me, we can go together. Okay, so once the first step is to check that we have the GPU connected, okay. So normally Colab is going to give you a GPU for free if you have not used it today, because they have a limit. So once that you have the notebook connected, you can read like all the cells, okay, but it's just like install. The first step are to install BiaPy, okay, so we can play the installation, okay. We can go forward. So now here there is an option to mount your files or your data set, okay. You can play these cells to use your local files, but we are not going to use this option. We are going to use the third one, okay. I'm just presenting them so you can see, but we are going to use one data set that is already prepared for this notebook, okay. You can also not only from your local files, but you can also mount your Google Drive. From there, you can also fetch files or use files. Okay. So the third option here, the example dataset that we have is it's the dataset that was prepared for zero cost notebooks. We can just play to download the dataset. Once that we have the dataset download, we just need to put the path correctly. Okay, so in this case, this is just explanation, and here are the variables. Okay, so where is the training data, the training labels? Okay, then the test and the test data. Where is the test data? And then where is going to save the workflow, all the files that are going to be generated. Let's say in this case, the instances of the images, okay, of the test images. So we can play this cell. Are you following me, guys? Yes, no?
I am.
Yes.
Yeah, okay. Yeah, sometimes I feel that I'm just like speaking to the void, but okay, let's go. So then we can play this cell to visualize some data examples, okay? Samples. We can maybe wait
Is the
to
installation
play it.
already done, Danny, in your machine, or not yet?
No.
No. So that's the part that takes usually more time. It's like 3 minutes or so, no?
Yeah. Sometimes it's less, sometimes not. I mean, because Collab is being like upgraded every 2 months or something like that. So sometimes more packages are required. Sometimes they have already all our requirements. So it's like that. So, but yeah, it's taking a bit, but okay. The rest of the cells uh, are not going to take that a lot of time. So Let's see if the installation is there. Well, we can go ahead maybe. No, but I want to wait until this cell is, so I can play with that. So everyone is also waiting. Yeah.
Yes.
Yes. Okay. Let's see. Okay, I have already installed everything. Okay. Yeah. So this is the cell. So here we have we can display the images within the data set that we have choose. Okay, we have not only here the training set but the test set. Okay, so you can here visualize different images of the data set. Okay, this is the raw image and this is the corresponding labels. Okay, so if we continue here, there is a brief description of how BiaPy resolve this problem. So it's a more maybe more detailed for I mean for those that want to deeply know how this problem is resolved, you can go to the documentation. But briefly, we are going to here learn a few representations, okay, and then we are going to fuse them in a post-processing step so we can recover the final instances, okay. So apart from that, there is also a cell dedicated to the bioimage model zoo. This, if you play this cell, it will find all the models in the bioimage model zoo that BiaPy can consume, and specifically for this task, for the 2D instance segmentation task. Okay, so whenever you are using a data set that is close to one of the models, maybe you can use one model, one pre-trained model, so you can accelerate or speed up the training process. Okay. Also, this notebook is prepared for training and testing, but we have also a notebook dedicated just for inference, and you can also use these models to just infer in your own data, okay, without any fine tuning. So you can do both things. So after that, we need to play this cell that will tell the notebook whether we are using a BiaPy model, or if you are using a pre-trained model from the bioimage model zoo, you will need to copy paste the animal name, okay, here in a field that is going to appear here, okay. But in this case, we are going to use a model from BiaPy, okay. So just select that source, and this is maybe the part where you can customize more the workflow. Okay, so these are a few variables that you can tune. You can I mean, you can just play the cell, and with the default values it will work. Okay, but here for the sake of the example, we are going to reduce the number of epochs here. Okay, so we have, well, let's explain a little bit all these sections. You have like the name of the model, in this case my 2D instance segmentation, okay, whether you have ground truth for the test data or not, because maybe you don't have it. So if you have the predictions will be compared against this ground truth, and you will have like some metrics to see how the model performed. But if you don't have it, you can just unclick it. Then for the training, we need a validation data, so we can prevent the model to overfit too much in the training data. Okay, so we normally put this like 10% of the training data. And then we have like a few basic parameters and then a more advanced parameters. Okay, so to not, as we don't have much time left, to not overwhelm you too much, we can just leave it like that. I changed the number of epochs to 20. We can leave the paging also to 20. And for advanced, we can just leave them as they are. Okay, so I will just play. No, it's true that to speed up a little bit the process, we can use the learning rate scheduler one cycle. Okay, to speed up the learning process in this case. Okay,
And maybe change the learning rate, no? Or no?
Oh yeah, true. Yeah.
Reduce it a bit because
Yeah. It usually works better with one less removing 1 0. Okay, so we can play the cell again, and then finally we have the cell to play to train the model. Okay, so if you play it, it will start running, and it will first train the model and then test with the test images that we have provided. Okay, so it will take a bit of time. So I will explain you the rest of the cells that you have here. So once that it's been done, you will have a cell here to show the training plot, how the loss was the error of the model is going down. Okay, this is the thing that we expect. Okay, so and apart from the loss, also a metric in this case we are measuring the intersection of a union. I mean the overlap between the predictions and the real ground truth. Okay, so you see that with more epochs, the model is increasing this value. Okay, to a more to better IO. Okay, so apart from that, also you have a cell to visualize the results that you will get. Okay, I'm not playing these cells, so I can have this to explain to you, but you can play the cells. Okay, and you will see the results. Also, there is a cell to display where are the output images. Okay, in this case, this is the path, this is the job name that we have selected, and then on the results the same name, and then in per image instances you will have access to the previous instances out of the test images. So all these paths and all the further explanations are explained in the documentation, in our documentation, so you can find more information there. Okay, so I want to, apart from these cells, to download the results. Okay, to download the training models, the training weights, the configuration file that you have just used in case you want to share with someone else the thing that you have just trained. Okay, I want you to also dive into this cell, which is another cell specifically for the bioimage model zoo, to export the model that you have just trained, given some fields that are required to create the package of the bioimage model zoo and the format. Okay, you will have a zip of your model that you can upload it to the bioimage model zoo website. Okay, and that's all. Yeah, so after that there are more advanced options that I'm not going to go in more detail here because we are running out of time, but you can play with them also to fine tune or to touch some thresholds that are in charge of doing the instances out of the raw predictions of the model. Okay. So with these last minutes that I have, hopefully, well, yeah, if you were following me, you will maybe have a chance of having these cells, the output of these cells, okay, and check by yourself how the model perform in the test data. But I want you also to show that apart from these collab notebooks, as I explained, we have um, more ways to run BiaPy. Okay, so I want to briefly show you the graphical user interface, which if you go to the landing page here in the first tab, you will find a button to download the graphical user interface, and you will have I have downloaded already the binary, and once that you run it, you will have like something like that. And this is the graphical user interface to do the same thing that we were doing that Nacho was explaining. You have here like links to the documentation, to our projects, to the forum, to these notebooks that we are running. We have templates, okay, and we have here two main checks: whether you have GPUs, whether you have Docker installed. It requires Docker to be installed. There is also a section with this tab here, this button here to to go to the wizard, so you can create the jam configuration file that you need to run your workflow. Okay, and in this tab here, in the tab of run workflow, you can select once that you have created your jam configuration file through the wizard, or maybe if you have yours, you will have you can load it in the GUI, check the consistency of the file, and then run the workflow. Okay, so yeah, I wanted to just mention this, that we have like more ways of running BiaPy. Okay, but I don't want to overwhelm you too much. So in case you have any doubts or anything to comment, please, even if we don't have like much time now in this presentation, please reach us out, okay? And we will be happy to answer any kind of doubts or questions, or help you tackling your specific problems. There is a question there.
Yes,
Do you
we have
segment?
segmentation pipeline supporting sparse annotations.
Yeah. So we have, yeah. It's in the documentation. We have a section to explain it, to explain in instance segmentation, but I mean it will require a few variables to touch. So I can maybe, if you can write me, I can guide you how to do it. Okay. But
Then
we can do that.
yeah. There's also a
So
question of what the input requirements are for the data.
the input requirement, what do you mean? Like
Yeah, I guess formats and
formats?
whether
Yeah,
we support or not in terms of dimensionalities and all of that.
yeah. So as Nacho has explained, we can handle 2D and 3D images. We support the most common extensions like PNG, GPA, I don't know, TIFF, this regular image extensions. Okay, and also we can handle, as Nacho has explained, SAR and H5 formats, and also not only one channel but multi-channel. So yeah, we I think that we cover quite extensive range of file formats, let's say.
Yeah, maybe it's worth saying that the only thing that we expect from the data is to be in folders. That if you have data that is paired, meaning we have one folder with the file, the raw data of the different images, and then you have for training the segmentations in another folder, then the names should be similar or in the same order. We don't force to have exactly the same name, but it would help if they are in the same order. Okay.
Yeah, we
And
have
nothing
that.
else. I think I don't think we have any more requirement like this.
No. And I want to say, just to add something on top of what Nacho said, is that you can have maybe here I will share with you a link to our documentation with the data structure that we require. But this, as Nacho was saying, everything like within the same folders, and yeah. Also the images, the names should be like paired. I mean, this is just an example, like training 001. Okay, you can have like whatever name suits you best, but it's true that the name of the raw image and the label should be like, the same. So they are like shorted in the same way, and they match each other. But apart from that, yeah.
Someone is asking if the size of the images for the training should be a specific size or recommended size.
No, not really. I mean, depends on your computer. If you can, for example, run BiaPy in a cluster with a big RAM memory, you can scale it up as you want. I mean, so yeah,
Yeah,
it
also, I
doesn't
mean,
matter.
I think it's more related to the annotation effort, no? You are able to annotate all the data that you have, and then you can store it in SAR files, and it's great. You can use all of that. But depending on the problem, you may need just a small annotated data set. Why not?
Okay, so I don't know if there are more questions. I know, as said, you can send us like whenever you want any message with your doubts, and we will be happy to answer them.
Yeah, absolutely.
Can I ask
Thank-
the last question, please?
Yeah, of course.
Thank you so much for the workshop. I was wondering if you had also recommendations for the labeling tools, the annotation tools. Do you use something particular?
It's true that we normally don't label too much because we are when we collaborate, we normally are in part of running the models and these things. But I do have some recommendations. I would suggest you using Napari maybe, because yeah, maybe the labeling in Fiji, for example, is not like very good in my opinion. So yeah, I would use Napari, but depending on the case, I mean, I don't know. Here in my lab, for example, they use like CADMate for doing the skeletons and of neurons. So I don't know. Depends on your specific task, but yeah, Napari is a good approach.
But so essentially, you clearly always need like either a binary or a labeled image. You cannot work with, for instance, objects. Like you cannot give it a JSON file with Yes, you
With
can. I see Nacho nodding.
but you mean like the
Like
skeleton, so like or
no, I mean like the description of the polygons. So because there's some software where you don't,
Mm,
if you label, you don't generate like a labeled or a binary image, and it's also more memory efficient. But I don't know, like, so in your case, do you really need to start from ground truth with labeled or binary images?
Yeah,
Is
yeah,
that
yeah,
a
yeah.
necessity? Yes. Okay.
Yeah. I mean, yeah, if you have like this kind of labels in other formats, you need like to convert them into
Okay.
somehow like these labels, images, TIFFs, PNGs, whatever, and then use them. Yeah.
Yeah, only for object detection, we use CSV files with the coordinates. So that's
With the center of
that's
mass
that's
of each blob
Okay.
that you want to. Yeah,
Makes sense. Okay. Thank you.
you're welcome.
No, thank you all really for being here.
Oh, there is
Pleasure.
another one. There is another
Oh.
one.
Okay, if the set is changing shape over images, would you recommend to provide different types of ground truth annotation?
Oof. Depends. It depends. How they change, how drastically they change. I mean, because whenever whenever you are playing with a data, it will have like some heterogeneity, right? I mean, if you have blobs, no, the blobs are not like always shown in the image in the same way, right? You have like some kind of a range. But if they change a lot, maybe you will need another class to identify them, or maybe it's just a matter of if you mean like changing, but if they are growing, but you can maybe resample that image or scale that image, like downsample it, something like that, so you can have a shape like it's more closer the to some blobs that you have been using for training the model. Maybe that could be an option. I don't know. Depends on I need to look at the images so I can answer you without I can provide you a better answer. I mean, because now I don't know. Depends on the task, I would say. From round to elongated. Well, if the shape, yeah, if they are like always in the same kind of size, it doesn't matter if they deform a little bit. We also normally, when we train a model, we normally apply like elastic transformations to like mm, do this morphological transformation. So the model, it's like can generalize a bit more.
Yeah, also, if you're doing segmentation, since as Danny explained, we are dividing that problem into the problem of detecting first the borders, maybe of the images and the inside part with some maybe directionalities or distances, etc. So maybe the data is still good if the borders are properly defined and you don't need to annotate more. But this is totally experimental. Until you see your data run through the workflow, it's hard to say.
Yeah. There is another question. Does the input for 3D segmentation need to be isotropic? No. You can work with isotropic and isotropic data, whatever data you have.
And very important, we have solutions for anisotropic data. So
Yep. Yeah. You can input in these configuration files that the BiaPy needs. You can put there your data resolution. So some of the post-processing and per-processing are taking that into account, so they can do a better job. So okay, so Instagram normalization. We have Instagram normalization, yeah.
yes.
And we have also Clage. Super, thanks. You're welcome.
Okay, how to choose different learning rate schedules. So here is a fight that we have, Danny and I, because I'm a one cycle person and he's more
Yeah. Probably you notice it when I was in the notebook and I was like changing the learning rate, right? The learning rate. Because I'm not very used to the one cycle thing, and Nacho is more on that. I normally use like warm up cosine decay.
Yeah, me
Uh,
too.
so yeah, I normally put the learning rate always like if I'm using that W as 0001. And then yeah, Adam W, and then warm up, I don't know, a few epochs, and then like slowly, cooling it down.
Yeah. For prototyping, I use one cycle a lot because very
Mm.
fast you can get results that make sense, or at least you see if you are in the right direction.
Yep.
And then for the notebooks, because usually they have to be very pedagogical, then we use one cycle because it's fast to train, and then you see like we have proper segmentations of cells in 10 minutes. So
Okay, yeah.
Nicolas, you have a question?
Yes, thank you, Daniel. Do you have a method to select the subset of images that you're gonna use for annotation and training? Meaning, do you have a way that takes into account either like intensity metrics or something like that to take the most variable subset? Or do you do it just like you just pick by chance like a few images and then you use those and annotate those?
Uh, yeah, so we don't have something specifically for that, but what we do have, but I don't know if it's very related to your question, is to add like some filtering. So to just take patches, whether you have like label information or not, or whether, I don't know, maybe you have a huge or large image with just something in the middle that you want to capture, right? The rest is just background or something like that. So maybe based on some intensity, just let the model train just with that part and discard the rest. All these things we have that implemented,
yeah, but
but
we don't
we
do
don't.
the so the annotation has to be already done,
Yeah,
and then
yeah,
we
yeah, yeah, yeah.
select samples that are good or better based on what we observe in the intensities.
But do
But
you
we
see what
don't.
I mean, Nacho? Where do you have a method to find the most? Because what I see a lot of image analysts doing, and we somehow do that in our facility too, is that we
Mhm.
take them sort of by chance or informed by the user plus by chance, like just, okay, random subset to prototype the ground truth, let's say. You see what I
Yeah,
mean? And I wanted to know
yeah.
if you did it by chance too, like random, or if you were informed by some sort of metric, because we're trying to do that
So what we
here at
do,
the
we have
facility too.
those as Danny said, we filter the samples that are annotated once they are already annotated. If you
So
go
you need to
a
annotate
step,
them
step,
first.
yes,
Mhm.
but if you go to the previous step when you have to annotate, usually we follow usually Danny and I when we collaborate these kind of protocols where we talk with the people, we look at the data, and then we decide which areas need to be annotated or not. And sometimes I don't know, we have a project where we have to detect every single cell in the brain, and then we are like, okay, then look, the cortex looks very different from this other part, so take some subvolumes from here and there, and then let's start by doing that. But we don't have anything automatic in BiaPy to do the annotation because BiaPy already assumes you have the annotation.
Hmm.
Yeah.
All right, thank you.
But we normally do that if we are like collaborating with someone that is producing the annotations in this kind of cycle, like they annotate some images, we train the model, we pass like some unseen images through the model. They will have like some, like not perfect labels, and then they can refine that and add them to the training data, uh, retrain the model again, and then the model will get like better and better until you have like something. So yeah.
All right, guys. Thanks.
Okay, maybe we should wrap up.
Yep.
Thanks a lot, everybody. And
Yep.
as Danny said, we are always here, so I'm very happy to hear from you. Okay,
Bye, guys. See
see
you.
you.